This protocol allows the production of safe and efficient nanoliposomes, which can be used as transfection vehicles for therapeutic messenger RNA. These nanoliposomes facilitate mRNA uptake in cells, resulting in increased protein expression levels without affecting cell viability.
The main advantage of this technique is fast and easy generation of stable nanoliposomes with defined size and homogeneous distribution. Furthermore, they protect the encapsulated mRNA from degradation and lead to prolonged translation.
Demonstrating the procedure will be Antonia Link, a PhD student from our laboratory.
To begin this procedure, prepare stock solutions for the lipids DC cholesterol and DOPE by dissolving each in chloroform to a final concentration of 25 mg per milliliter. Mix 40 microliters of the dissolved DC cholesterol with 80 microliters of the dissolved DOPE in a glass flask. Vaporize the chloroform under an argon gas flow for 15 minutes. Next, fill a desiccator with silica gel. Place the open glass flask inside and apply a vacuum overnight to ensure that the remaining chloroform is evaporated. The resulting lipid film will form inside the glass flask. The next day, add 1 milliliter of nuclease-free water to rehydrate the lipid film and vortex the suspension for 15 minutes. Place the suspension into a sonication bath for 1 hour. After this, assemble the mini extruder according to the manufacturer's instructions. Fill a syringe with the liquid.